So for this video, we're going to look at muscle fiber cells and the contracting sarcomeres inside of there, and we're also going to look at nerve impulses a little bit. So what we first need to look at here is the actual outside of the muscle cell. As you see coming out of this little picture here, this long strand coming out right here, that is a single muscle fiber coming out of what we call fascicle, which is fascicles wrapped in a paramecium. And these muscle fiber cells are wrapped in endomecium. So on the right underneath there, we have what's called the sarcoplasmic reticulum, which is another name for the plasma membrane. And then coming out of your muscle fiber cells, or within them, I should say, you have what are called myofibrils. And those myofibrils are what contain those contracting units called sarcomeres. Right now, those sarcomeres are going to be in charge of contracting, and the more we contract, the smaller that muscle gets, and the stronger that contraction will be with the activation of them. We have something that's called an I band, which is going to be the area outside of what we call myosin, which you'll learn in just a minute. And then you have the A band, which is the darker band area, which is where you see this blue line running through there. It's just darker or thicker filaments, which we call myosin. So you have an A band that goes from one end to the other. And then you have your I bands that are outside of that. This over here, whoops. Over on the side, on the other side of the A band, you would have another I band over here. Okay. So you have an I band on one side and I band on the other. Okay. So when we zoom in on those myofibrils, you can see they're all stacked. Myofibrils are inside of your muscle fiber cells. So this whole thing is a muscle fiber cell. Your myofibrils have these things called sarcomeres. You can see you have the I band and then your A bands. Okay, and they'll just keep alternating, and that'll go on the entire length of your muscle cell. And one other word you're going to hear is the sarcoplasmic reticulum, which is where calcium is stored, and that's going to be important in changing how myosin heads can actually attach and cause contraction. Here's another zoomed in look. Once again, you can see as we move down the myofibrils, you see more and more sarcomeres. Those are your contracting units. So the more you have, stronger that muscle is. And then we get inside those myofilaments, and you have inside of those sarcomeres. Sarcomeres is the contracting unit. So a sarcomere in this picture would go from, you could say from this side all the way over to that side. Okay, that's a sarcomere for us. It includes actin and myosin. Myosin, what they use is ATP in order to detach their heads and extend and pull closer those actin filaments, right? So they have myosin heads are on the end. So all of these little things sticking off here, those are little myosin heads, and this whole blue is the myosin. And they form what we call cross bridges, which just means they attach to the actin to pull it closer. And then the red in this picture is what we call actin. And those are the things that actually slide. So myosin actually never really moves. Myosin stays put. And what they do is they pull this actin closer. And the more of these sarcomeres that are pulling actin closer, smaller that muscle fiber becomes and the stronger that contraction becomes. So you have myosin, which are thick bands, heavier, they're darker. That's your A band area, right? So from here, whoops, to there, that would be your A band. And then your I band would be out on the side. So that's an I band. And there's an I band out on the side over there, okay? And like I said before, you have a sarcoplasmic reticulum that wraps around the outside of these myofibrils. They store calcium until a nerve impulse comes and it causes it to then contract, okay? Here it is again. Here's your myosin is in green. Right here, you have your myosin heads that are creating cross bridges with your actin. Your actin is your purple in this picture. Right? Remember, myosin doesn't move. but the actin will contract, or the actin will slide closer together during contraction. Okay, here's a couple different pictures of what's really going on. Here's your myosin heads. So as one reaches up and grabs, another one is kind of grabbing and pulling, and then that first one will detach and kind of move. And that's how you consistently have this muscle contraction when needed. Here you can see it when it's relaxed. Here you can see it's contracted, right? When it's relaxed, you can see that we have some space with this I band and what we call this H band. We don't really have to worry about the H band here. But when we contract, that actin slides closer, and notice how much tighter that space became, just like how much tighter this space over here became as well. Okay, everything slides in nice and close, meaning those actin are going to slide closer to each other. Okay, how do we get our muscles to actually contract? Well, we need a nerve impulse. So all of our muscles have nerves that are attached. So when we say we want to throw a ball or we want to move our muscles, the nerves, our brain sends a signal down our nervous system, first down our central nervous system, then to our peripheral nervous system, and that causes muscle contraction. And we're going to get into the specifics in the nervous system, but when we look at a motor unit, we're talking about one neuron and all the muscle cells it stimulates, right? So let's fast forward really fast. Here's one neuron coming down. Here's one motor unit coming down. Here's a second one coming down. Now notice they kind of branch out. So one neuron may stimulate multiple muscle cells, muscle fiber cells to activate, right? And then this motor unit 2 is going to activate others. And the more motor units we activate, that means the more muscle fibers we activate, which means stronger contractions. So that's a motor unit when we take the nerve and the muscles that it is causing contraction with, right? And when we look at this neuron, when it comes down and it connects with that muscle, we call that a neuromuscular junction. So here's the end of a neuron right there, okay? And let's say this is our muscle. What's going to happen is we get a neuromuscular neuron and muscle junction. And what happens is we pass these things called neurotransmitters, which are all in here. These neurotransmitters get passed across. So that's the chemical signal that gets passed across. And then when receptors receive that, that then causes calcium to be released, and it causes something to happen with the myosin and actin, which you'll see on a video. Okay. So we take this signal, we pass it down the neuron. Those neurons are going to hook up with a muscle, making a neuromuscular junction, and that's going to cause those sarcomeres, those muscles, to start to contract. And the myosin heads will reach up, grab those actin, and start to pull them closer to each other. Okay, we'll talk about action potentials when we get to the nervous system, so we don't have to worry about that right now. And the last thing is the sliding filament theory. Okay, so when those neurons fire and it causes the muscle to start to contract, what's really happening inside of there is calcium, which is stored in the sarcoplasmic reticulum. Calcium is released from the sarcoplasmic reticulum. And what it does is it allows those myosin heads to attach to actin. Here it says calcium attached cross bridges to actin. What we mean by cross bridges is the myosin heads are now able to reach up and latch onto that actin to start pulling it. If we don't have calcium stored in the sarcoplasmic reticulum, then you're not going to have muscle contraction. That's going to cause some issues. So the nerve impulse comes down, it causes calcium to be released onto or into these areas where actin is. That changes the shape of actin a little bit, along with some other things, and allows for myosin to then attach that actin and pull it closer. And we call that the sliding filament theory because the actin is sliding closer to the other actin, and in between, you would have those myosin. Okay, So you can see down here in this picture, you have a relaxed, well, you have actually two relaxed sarcomeres. They're side by side, right? You can look at the I band and the A band, right? And over here, you would have another A band right there. That's gonna be, whoops. That's another A band. Right? And as you see, as it starts to contract, you can see how much closer those act and slide to each other.